Hello and welcome to Anonymous Med. This is my second lecture on hematology. So let's begin. So first of all, we're gonna learn about hematopoiesis. Hematopoiesis, as the name suggests, it is the beginning or the starting of formation of hemats. The all the blood cells formation is known as hematopoiesis. So it starts in the yolk sac. This can be a potential MCQ. It starts in the yolk sac in third week of intrauterine life. Then in the third month it occurs in the liver. In fourth month of intrauterine life, we are talking of intrauterine life, it occurs in liver and the bone marrow. And just before birth, the hematopoiesis actually occurs in the bone marrow, and then till the adulthood it lasts in the bone marrow. Now, in adults, the hematopoiesis occurs in the flat bones like ribs, sternum, and pelvis. Now the basics of the hematopoietic stem cells. These are the hematopoietic stem cells present in the yolk sac in the intrauterine life. So these cells migrate to the different parts of the body, like first liver, spleen, and bone marrow. So and then in the adulthood, it appears in the bone marrow mostly. So the hematopoietic stem cells are basically of two types, majorly of two types: the colony lymphoid forming cells and colony myeloid forming cells. So the colony lymphoid forming cells and colony myeloid forming cells. Now the lymphoid forming cells just forms the lymphoblast, as the name suggests. Lymphoblast is formed. Blast means immature cells. Now the myeloid forming cells are myeloid potent cells are further classified in four categories: the erythroblast, the RBC immature RBCs, the megakaryoblast, the immature platelets, the myeloblast or all the WBC cells, the granular types, and the monoblast. Monoblast is considered, as we know, the WBCs are further divided into granular and agranular. Under agranular, there comes the monocytes and the lymphocytes. So out of which the lymphocytes has been derived from the colony lymphoid forming cells, and the monoblasts are actually formed from colony myeloid forming cells. So these are the four types: the erythroblast, the megakaryoblast, the myeloblast, and monoblasts. Now, erythroblast, of course, they forms the RBC. Megakaryoblast, they form the platelets. The myeloblast, they form neutrophils, eosinophils, basophils, and monoblast, they form the monocytes. Lymphoblasts, they form three types of cells: the T lymphocytes, the B lymphocytes, and the natural killer cells. Remember this, the natural killer cells are actually formed from lymphoblasts, and as we know, the T lymphocytes and B lymphocytes are the functional classification of the lymphocytes. The other type can be small lymphocytes or large lymphocytes. That is the other classification. Now, moving on to potential MCQ, just I have written here. The marker for stem cell, hemopoietic stem cells, is CD34. It can be a potential question. The marker for hemopoietic stem cells, it is CD34. Moving on to the WBC disorders. For the WBC disorder, we should actually know the basics first, and today I am just dealing with the basics. The actual WBC disorders will be dealt in next lectures. So first of all, we are looking for the myeloid series, these all, but we are potentially looking for neutrophils since we are talking of WBCs. So let's just talk of WBCs for now. So, and as we all know, neutrophils are the most found, so that is why we are looking for neutrophil series. So first it is myeloblast. Here, as you can see, the myeloblast form the promyelocytes. The promyelocytes, these are the largest cells. Then it forms the myelocytes. Promyelocytes form the myelocytes. Now moving on to the next slide. Now, the myelocytes form the metamyelocytes. Myelocytes form the metamyelocytes, the promyelocytes, the myelocytes, the metamyelocytes, and then it forms the band form. Up till metamyelocytes, the lineage is just the same, okay? And after, since it's forming neutrophils, there is formation of band form. Just the name you have to remember. Okay, now finally the neutrophils are formed. Now, normal TLC count, the total leukocyte count, is 4,000 to 11,000 per cubic mm, and the WBCs, again the classification, as I told you, the granular types and the non-granular types. neutrophil, basophil, eosinophil, monocytes and lymphocytes. These are the basics we have learned from 9th grade. Okay, now moving on to how to identify WBCs in peripheral smear. The P/S means peripheral smear in short form. So identification of different WBCs in PS. So first of all, this is the basic PS we can see in almost all types of uh, peripheral blood smear. So these small cells, clumped cells, are known as the platelets. They appear as aggregated group and very irregular and very small. They will be very lesser in count. I did draw a lot of them, but they are less. Now moving on to the RBCs. RBCs, as we all know, biconcave oval shaped structure and with a paler in the middle, 1/3 almost of the total size, central paler. Now moving on, this is eosinophil. Eosinophil will actually look like this, as there is brick red cytoplasmic granules. The brick red cytoplasmic granules, you can see, this is eosinophil. Next up, which is neutrophils. Neutrophils, as you can see, the eosinophils and the neutrophils, their sizes are almost equal. The neutrophils, the eosinophils, the sizes are almost equal, whereas the lymphocytes, as you can see here, the lymphocytes which I am looking here are the small lymphocytes, so they are almost in comparable size with the RBCs. Now next up, that is the basophils. In the basophils, it will be almost covered with the coarse granules, the dark blue granules, and you cannot actually clearly identify the nucleus in it. And here the neutrophils can be 3 to 5 lobed in neutrophils. I forgot to tell you this. Now moving on to monocytes, the largest cells. These are the largest cells, the monocytes. Monocytes are the largest cells and have kidney-shaped nucleus. Almost 50% of the cytoplasm is covered with a nucleus. You have to remember this too. Now, this is just a short classification for non-neoplastic disorders of WBCs, the non-cancerous disorders of WBCs. It can be leukopenia, leucocytosis. Penia means decrease in WBC and cytosis means increase in WBCs. Again the same, mostly it is neutropenia, neutrophil neutropenia. So, and reactive proliferation of WBCs can be cytosis, leukocytosis. Moving on to the next slide. Now we are looking for the causes of leukocytopenia. So in leukocytopenia, there is decrease in WBCs. Causes can be two, just have to remember this. Aplastic anemia, of course, aplastic anemia, it is pancytopenia, where almost all lineages of the cells are being decreased. So yeah, aplastic anemia will be there. Another can be drug toxicity. Now, agranulocytosis, that is either lymphocytes or the monocytes. It is clinically significant, and there is decrease in WBC count. Simply, just remember this much. Now, moving on to the different types of WBCs. That is the first one, that is neutrophil. I have actually written it in an order. So, first the granulocytes and then the non-agranulocytes. So, in the neutrophils, increase in neutrophil count is called as neutrophilia. We are actually moving on to the cytosis. We are looking for the cytosis, the increase in WBCs. So first can be a neutrophilia. In neutrophilia, which is increase in neutrophil count, the normal neutrophil count can be 40 to 70% of total WBCs, the TLC, the total leukocyte count, out of which 40 to 70% should be normal neutrophil count. But if there is more of this, so we'll call it as neutrophilia. Now the causes of neutrophilia, very basic, that is acute infections. Acute infections, remember the word acute. Whenever you hear acute, you should actually go through whether it is acute inflammation or acute diseases, infections, it is always the neutrophils which is increased. But if they mention chronic, so you have to actually think of lymphocytes. Whenever you hear the word chronic, you have to go through, as you can see right here, chronic infections, it is lymphocytes. Just remember this basics, like chronic inflammation, again lymphocytes, and chronic inflammation, acute inflammation, it will be neutrophils. The next example could be tissue injury. Yeah, tissue injury, again neutrophils will be activated, so their count will be increased. The bacterial infection, again remember this, the bacterial infection, it is actually due to neutrophil increase. And for viral infection, you have to remember the lymphocytes. Just remember, it can be a potential MCQ. The myeloproliferative disease, again CML, like CML. In CML, this is chronic myeloid leukemia. In CML, neutrophil can be increased. Now I will tell you very nice thing about CML later on. Okay, now the next step, that is eosinophilia. In eosinophilia, again there is increase in eosinophil count. The normal eosinophil count is 1 to 6% out of total 100 leukocyte count. In 100 total leukocyte count, there will be 1 to 6 eosinophil present. The causes of increased eosinophilia, if there's increased count in eosinophilia, it's known as eosinophil. So eosinophilia. So in eosinophil count will be increased in allergic reactions. This is really basic. We have learned it in 11th, 12th grade. So in allergic reaction, eosinophil will be increased. Very easy, remember this. Or parasitic infection or helminthic infections, remember this. Here was the bacteria, here is the virus, and here in eosinophilia, parasites or in helminthic reactions, the eosinophil count has been increased. Or type 1 type of hypersensitivity reaction. I will give you another video on hypersensitivity reaction, and type 1 is actually known as anaphylactic reaction if you have learned it from your basics. So in anaphylactic reaction, again it is an allergic reaction, that is why eosinophil has been increased. And tropical pulmonary eosinophilia, it is again a disease. I will deal it with later on. So moving on to basophilia, this one is interesting. Just one case you have to remember here, that is chronic myeloid leukemia. Here, this one, chronic myeloid leukemia. This you have to just remember this. When I was in first year, my sir once told me and asked actually, where do you see chronic myeloid leukemia? In which type of cells will be increase in chronic myeloid leukemia? So the answer was basophil, and from there I actually remembered. Earlier I wasn't clear with this, why is that, but just you have to remember that in basophilia in basophils, when basophils are increased, the case will be chronic myeloid leukemia. Definitely it will be chronic myeloid leukemia, no other thing you have to remember about this. Easy. So the normal basophil count will be less than 1%. It can be present in a blood smear or it can't be. But if it's seen and in more than 1%, so you have to actually think of chronic myeloid leukemia. Very simple. Now the last slide, very simple one again. So the last slide. Yeah. This is lymphocytosis. Lymphocytosis, again, increase in lymphocyte count. The normal lymphocyte count is 15 to 40%. 15 to 40% will be in a normal count of lymphocytes. If you see more than that, it will be lymphocytosis. Now, the causes of lymphocytosis, very simple. You have to compare the neutrophilia with the lymphocytosis. Tuberculosis, again, it is a chronic infection. This one, chronic infection. Acute infection was the neutrophilia. So this is lymphocytosis. So here will be tuberculosis. You have to remember this. This can be a very potential MCQ and a good point to remember. Very easy. Just remember tuberculosis. It is a chronic infection. Viral infection. In neutrophilia we have read the bacterial infections, and in eosinophilia we have read the parasitic or helminthic infections. The final one, which is lymphoproliferative disorders like CLL, that is chronic lymphoid leukemia, or ALL, acute lymphoid leukemia. Again, wherever you hear the chronic word, you have to remember it is for lymphocytes and acute for neutrophils. Just a little difference here. For acute ALL you have to acute lymphoid leukemia. You just have to remember it's for, again, here the name itself suggests it's lymphoid. So lymphoid, it's definitely will be lymphocytosis. Okay, moving on to the last part, that is monocytosis. Monocytosis will be increase in monocyte count. The normal monocyte count is 2 to 8%, so you'll see 2 or 8 out of 100 in a blood smear. So causes, now this thing you have to remember, malaria. M for malaria, M for monocytosis. So monocytosis, the only M standard WBC. So it is seen in malaria. Wherever you see large number of monocytes, just think of malaria. Second can be endocarditis. Endocarditis could be there. Rickettsiosis. I'm not so clear with this, I'm sorry. Rickettsiosis, yes. And viral infections, again, the viral infections here. viral infections in the lymphocytes. You can just remember that viral infections actually increases the A granulocytes. Both are A granulocytes, so viral infection increases the A granulocytes. That's all for today. Thank you. I hope you enjoyed the video.